And thank you again for joining today. Welcome to the first session on bioinformatics for infectious diseases. In today's session, we will speak about the emergence and development of bioinformatics in infectious disease research. We will discuss what infectious diseases are and how they can be characterized from a data perspective, specifically next generation sequencing data. We will also show several examples for bioinformatics analysis methods that are used in pathogen diagnostics, viral evolutionary studies, antiviral drug discovery, vaccine design, as well as the characterization for emerging drug resistance. To do so, we will rely on the T-BioInfo analytical platform designed for virology studies. For those of you who have still not completed their registrations, you will also get a good overview of the OmicsLogic Infectious Diseases Bioinformatics Training Program. Interested in effective utilization of bioinformatics for infectious diseases, immunology, and microbiology. For all of you joining us for the first time, my name is Deepsha Biswas, and I am the OmicsLogic Community Manager responsible for daily interactions with thousands of users that are part of our bioinformatics community managed by Pine Biotech. Over the past couple of years, we have seen participants from all over the world join our training programs, take online coursework, and utilize our big data analysis tools in various research projects. Let me start by introducing our company and collaborators to share with you details about the upcoming OmicsLogic Bioinformatics for Infectious Diseases program and explain how the training resources we will discuss today can be used effectively for research, independent learning, or as a structured training program. What we will discuss today and throughout this program is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. Under his leadership, the Tauber Research Center started collaborations with research teams in such universities as the University of California, San Francisco, Institute Pasteur, Tulane University, and many others where research labs are working on cutting-edge research projects studying viral and bacterial infections and designing novel drugs and vaccines to treat and prevent infectious diseases. Now, before we move on, I wanted to invite everyone to come and join two of our groups that we have on LinkedIn and on Facebook. So Dr. Mohit, if you can please share the links in the chats. So if you are new to our community, we would urge you to come and join these groups where we keep sharing updates regarding these programs, as well as any other updates from the industry. So please join us here. In the first session of Bioinformatics for Infectious Diseases, we will talk about next generation sequencing. To introduce the program to you, we have our program mentors, and let me start by introducing them to you. Dr. Harpreet Kaur is a certified OmicsLogic trainer and expert bioinformatician who will guide the sessions and assist participants with projects and analysis during the program. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is a OmicsLogic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is our co-founder and CEO at Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. I will pass it on to Ilya to talk about the increasing role of bioinformatics and data science in infectious disease research and introduce you to the types of challenges data analysis, data organization, integration, and visualization can solve for biomedical research projects. Over to you, Ilya.
Thank you, Deepsa. So let me start this by asking you a few questions before we begin. So the first question I have is, what is your current level of education? I know that we have here participants from all over the world, and what we are interested in is not just conducting these online programs, but also developing a long-term relationship with a community that is interested in application of bioinformatics in a variety of domains. So let me ask you this question. If you could please put your response in the chat box. So number one, what is your current level of education? 1A, high school. 1B, undergraduate degree. 1C, graduate degree. 1D, postgraduate degree. All right, great, excellent. Thank you very much. What we will do with this information is we will collect it and analyze it for our future programs. We understand that those that come from high school or undergraduate degrees need a lot more preliminary training, and so we will help guide you to that training so that you are familiar with the terminology and are able to do the analysis effectively. For those of you that are graduates and have postgraduate degrees, we welcome you to benefit from the more advanced materials, and so we'll help shape that curriculum for you, introducing some of the more advanced topics and projects for you. Let me ask you another question. What do you expect from this program? So this is the first session in our Bioinformatics for Infectious Diseases, and today we'll talk about next generation sequencing, what kinds of questions and what kinds of projects you can do with different types of topics that we will cover. So what do you expect from this program? By joining this program, you can learn about bioinformatics in general sense. You can get familiarized with infectious diseases and some of the challenges that are being researched there. You can get an overview of bioinformatics and virology. So a lot of people join this program to learn about COVID-19 and other viral sequences. And then 2D, apply bioinformatics to a research project. So if you already have a research project in mind, a data set, what would that be? Okay, excellent. So it's great to know that a lot of you are interested in a combination of these, and that's fine. So really what we're interested to understand is how well can you utilize some of the resources that we'll cover today to specific projects that you have a true passion and a drive and a motivation to address. So excellent. Thank you very much for answering these questions. Again, we will analyze this information for all of the participants of this program, and we'll be interacting with you. Some of you have seen, and you will see at the end of this session, that we have multiple options to register for the program. Some of it includes interaction with a mentor. And so this information is critical for a mentor to be effective in those types of interactions. Okay, so let's talk briefly about what infectious diseases are and how are they different from non-infectious diseases. An infection is essentially an invasion of an organism's body tissues by disease-causing agents or pathogens. Their multiplication and the reaction of host tissues to the infectious agents and the toxins that they produce. So the infectious diseases are also called transmissible diseases or communicable diseases, and they are the result of an infection. The pathogens include viruses, bacteria, fungi, parasites. All of them have subcategories, but all of them have the key pathogen that is causing that infection. That's why they are transmissible and communicable, because the pathogen could be spread between individuals. Non-infectious diseases are chronic diseases, so they cannot be passed on unless they have a genetic background. And they're caused by such factors as environment, nutrition, lifestyle, and genetic inheritance. The World Health Organization recognizes 4 main types of chronic and non-infectious diseases: cancer, cardiovascular, chronic respiratory diseases like asthma, or chronic obstructive pulmonary disease and diabetes. But we will also talk in the context of this program about many pathogens that actually contribute to some of these non-infectious diseases. So we will see that there's actually a good relationship between both of these, and it's not such a clear cut. Now, when we talk about these pathogens, one of the key ways that they harm the host is by replication. So the replication of pathogens in the host leads to a major interference that starts from membrane disruption, as in the case of viruses, and release of toxins, as in the case of many bacterial pathogens. Also bacteria, and they colonize different tissues, interfering with the function. And so many of these interferences include modification of molecular biological pathways that are designed to fend off intruders and signal to neighboring cells and organs of coming danger. And so you can see that here you can see this is actually COVID-19 replicating and infecting the host cells. You can see how much of that virus is coming out, which means that inside these cells, there's an active production of these virions. And here you can see how some of the viruses and pathogens are also introducing specific signaling of toxins that actually cause different types of cell death and apoptosis, including oxidative stress and other types of stresses. Bacteria oftentimes damage the membrane and also cause secondary passenger pathways to be modified and modulated. And so you can see that a lot of this leads to scarring and tissue damage. For example, some of the pathways are not even kind of what the virus or the pathogen causes. They are the response, but because the response is so strong, it can have a major impact, a long-term impact on the organs. So in the context of this program, we'll take a look at some of these processes, identify such molecular pathways, and try to understand the relationship between the host and the pathogen. Now, how can the host protect itself? The host can fight infection using their immune system. So here you can see a few GIFs that you can easily find on the internet. And these GIFs really show you how the immune system responds. So here, for example, you can see a white blood cell chasing a pathogen. Here you can see what those blood cells look like as they seek to identify different types of present antigens that they would be signaling that this cell is undergoing infection. And so some of this is related to the immune system. Here you can also see an example of bacterial growth and the result of penicillin. Now penicillin kills bacteria. This is actually produced by HHMI, this little GIF. And you can see that what it essentially does is it just breaks the membrane of the bacteria. So we will talk about some of these processes and some of the interactions. We know that the host microbiome has some antibacterial producing bacteria and viruses. We know that it's a complex system. And so the major way that the host can fight off intruders is the immune system. So we'll learn about that. And the major way that we can help get that immune system activated are vaccines. But we'll also talk about antibacterial and antiviral drugs and how do they interact with the different pathogens and how do the pathogens respond to some of these viral and antiviral and antibacterial treatments, developing resistance. So we'll talk about some of that, and importantly, how to identify such trends in data. Now, vaccines have been discovered a long time ago, as far back as the early 1800s. And today, of course, we hear a lot about the COVID-19 vaccine, but really we're talking about a renaissance in vaccine design and development, a completely new approach that is driven by molecular knowledge. Now, overall, vaccines and antiviral drugs like antibiotics, or antiviral drugs and antibiotics, have been very, very effective health measures, but they come together with other epidemiological control measures or health measures that were very effective in reducing the number of death incidents from these pathogens, these types of pathogens. So for example, you can see here a brief chart. This is the rate of death per 100,000 population per year. And you can see how that rate of death has been spiking around pandemics, but overall, what we have seen over the course of the 20th century, from 1900s to 2000, we have seen a major, maybe 10 or more fold reduction in death rates as a result of control measures and ways to disinfect, so reduce the number of pathogens in water in the environment that we live in. And you can see here, for example, the first use of penicillin, the vaccines introduced, and then the number of people getting vaccinated. So all of these measures have been very effective. However, what we know today is that in the 21st century, this trajectory essentially changed. We've seen a number of pandemics or epidemics that have caused us to lose confidence in the measures that have been so effective, right? So we've seen the flu coming from swines, the flu coming from avian flu, right? So H1N1, all of these deadly viruses like Ebola, and of course now we have COVID-19. And you can see how this rate of death from COVID-19 has been surpassing some of the other major causes of death that are not necessarily a result of pathogens. So it's very important to understand that this trajectory is now putting us into a new age for vaccine design, antiviral drug design, and antibiotic approaches to treatment that combine molecular data and analysis tools that allow us to have a rational approach to answering such challenges. And so what we'll talk about in the program and in today's session, the focus of today's session is going to be on next generation sequencing, because this major type of data has completely revolutionized the way that we understand pathogens. So up until this type of technology was introduced, our understanding of pathogens was mostly driven by our ability to observe in using a microscope how these pathogens reproduce. And a lot of that has to do with isolating pathogens and studying them in the lab. Now we know that many of bacteria and viruses are very difficult to handle and study in the lab and isolate, and also to look in the microscope. Obviously, viruses are very difficult to look at in the microscope. So the sequencing technology has really changed the way that we understand the genetic material that can characterize the communities that they live in and the way that they interact with the host. So here you can see a brief overview of what types of insights we can get from this type of sequencing data. So we can understand the population structure and evolutionary history. We can look at specific sequences for detecting and characterizing strains of these outbreaks. And we've seen in the previous pandemics of Ebola and the coronavirus that we are in right now, how this type of data has been coming out almost in real time, allowing the international community of scientists to start asking very specific questions about where is this virus coming from? How is it changing over time? What can we do to try to apply previous strategies that were successful to this current challenge? Of course, we are talking here also about other types of more in-depth questions that are not diagnostic, but they're really to understand the mechanisms that these pathogens utilize to infect and overcome host responses. And this is important because this basic biology, this basic science, has really driven a number of translational projects that help us make these vaccines and antiviral drugs and antibiotics much, much more effective, because we understand the logic and can design within parameters that save time and save effort, that is troubleshooting and kind of trial and error based. So as a result of this kind of data, this kind of genomic data, we now understand and can study effectively the interaction between the type of virus and the type of host response in stages. We can look at time course of disease progression by looking at viral modification and host response and measuring that host response in very specific types of data. We can also look at different types of variation at the genomic level to understand resistance to antiviral drugs and evasion from such treatment over time. That's also an important part of it. We can also really characterize how the process of disease happens, giving us insight into protein-protein interactions and routes for intervention. And here you can see also a characterization of the immune response over time. And really, this ability to characterize the response over time is, of course, very useful for precision medicine. So actually, infectious diseases is one of those areas of precision medicine that has been tremendously improving as a result of genomic data, and genomic medicine in general has been most effective in addressing such pathogens as we are discussing in this program. Now, it's not just the sequencing data, right? Together with the sequencing data, there are some other types of data that are important to incorporate to be able to ask such questions and answer them definitively. And so we'll look in the context of this program at omics data, different types of omics data or multi-omics data that includes DNA variation, host gene expression, measurement of protein abundance, composition properties of microbial communities. And all of these are very different types of data that we all need to know how to effectively study and mine for patterns to understand such host pathogen interaction. So let's look at a couple examples of how we can deal with data and what kinds of data sets we will be dealing with to be able to ask such questions. So the role of bioinformatics, which the name of this program obviously is Bioinformatics for Infectious Diseases. So the role of bioinformatics in being able to study viral and bacterial pathogens is to kind of put the infectious disease, specific infectious disease into the context of data. And so as researchers are able to access, process, analyze, and visualize these kinds of high throughput data, they are able to start asking questions and answer them definitively, making assumptions and testing those assumptions by evidence provided in the data. So you can look at viral genomics and evolution, immune response, and identify biomarkers that help detect those, and then also microbiome and infection, a relationship that is very fascinating because our whole view of the way that these pathogens kind of exist and live together in communities is changing because of this ability to sequence data and understand what is really going on in real life. So we'll talk about this in a second, but first let's talk about this one major way that bioinformatics is changing our understanding of the microbial world. So the original appreciation of microbes, as I mentioned, was a result of seeing these pathogens in a microscope. And eventually today, we have so much data about these pathogens that we're starting to ask completely different questions about the communities that they live in. And so today we have a realization that these are communities or whole organs that actually have not only a specific pathogen, but a whole community of pathogens that coexist and form the specific relationship with the host because of their composition. And so we can talk about things like the microbiome and specifically the virome as well, which is where we can understand all of the viruses present in a particular environment. So We know that the virome, the studies of the virome are in their infancy because it has only recently become possible to see the virome in large sequence data using bioinformatics tools that can detect relationships between viruses, despite its extreme nucleotide sequence diversity. So we're not talking about a single pathogen, we're talking about many variations of the same pathogen that is very dynamic, right? They're changing all the time. Now, the size of the mammalian virome is not even known, although our own cells are outnumbered about tenfold by our bacterial microbiome. The number of viruses may be even tenfold higher. And human feces alone contain about 10 to the 8th to 10 to the 9th viruses per gram, so billions and billions of viruses. And additionally, the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. So as a result, because of the differences in the virome and the microbiome in different communities, we see that interaction between different hosts, for example, animals and humans, can be particularly dangerous. And so one of the questions that we can ask is, how do these pathogens change when they go between different environments? One of the key issues here, though, is that viruses are not even alive in a traditional sense. So the sequencing data, an additional aspect of the sequencing data, is really leading us to ask very basic questions about pathogens. So we know that the way we categorize life is actually divided up into 3 large domains: bacteria, archaea, and eukarya. And under this classification system, viruses are not even a part of the tree of life. So they're certainly not alive in the classical sense because they're not capable of metabolizing or replicating on their own. But at the same time, we cannot just classify them as inanimate because they are built of biological molecules and contain genetic information. And so we see that they are in between kind of being alive and dead. And that problem is changing the way that we think of life in general. So it's really the study of infectious diseases using data is changing our understanding of life. So there is a debate about how to classify viruses. And specifically, this debate recently has been kind of heating up because of discovery of these extremely large viruses, such as Pandora virus. They're so gigantic that they can be seen with a light microscope and contain more genetic information than some bacteria. And so it has been proposed, due to some intriguing similarities in DNA sequences, specifically in the gene that encodes for an enzyme called RNA polymerase, that such large viruses actually constitute a 4th domain of life. And so here we can see some joke or a funny version of the tree of life where we have bacteria, eukarya, archaea, and then a 4th domain. Maybe it even includes aliens. So really, the ability to sequence such information and understand what is going on in the real environment where these pathogens exist is not only changing the translational aspects of antiviral vaccine design and antibacterial antibiotics and things like that, it's also changing our very basic understanding of life and the way that we can approach asking some of these very fundamental questions. So at this point, I'm going to pause and I'm going to make sure that we don't have any questions. So are there any questions about what we've done so far? I wanted to make sure that those of you that have joined the session for the first time, that you have already joined our communities like LinkedIn and Facebook groups that Vivsa mentioned. And for those of you that are already a part of this program, that you are starting to think about some of the ways that you can use sequencing data to answer questions that either are basic or translational about the nature and the function of these pathogens and their interaction with the host. So are there any questions that I can answer right now? Okay, so please keep in mind, if I am going through the slides and you have any questions that come up, you can write down that question in the chat box and we will make pauses for such questions and answers. And you're free to ask those questions either during that time or before that time. So when we have a pause, we can address any of the questions that come up. So let's talk about next generation sequencing. What is next generation sequencing? What kind of data does it produce? What can we do with that data? So the process of next generation sequencing is when we take a sample, a biological material, for example, spit, saliva, and we can then sequence the DNA or the RNA that is inside this biological sample. The problem is that in between getting the sequencing data and being able to ask the questions, we need to structure the data. And so we'll talk a little bit about the process of bioinformatics pipelines that allow us to structure the data and start making that data available to ask specific questions. Now, the amount of such data is increasing exponentially. Here you can see some information on viral sequences, and this information is located in a couple of locations. For example, we have the Joint Genome Institute that hosts by January 2018, we see here over 700,000 total viral sequences, or over close to 400,000 unique viral sequences, so a huge amount of data, over 13,000 complete annotated genomes of different viruses, and millions of sequences of viral genes and proteins. So a huge repository of data. And the important part of this is that this kind of sequencing data is available in both raw and processed format. So when we talk about sequencing, we'll talk about how sequencing is done, different formats of sequencing data, and remember, all of this data is accessible. So all of these repositories are available for anybody to have access to. And in the context of the program, we'll have sessions where we actually access this data and we start asking questions of this data using some of the analytical tools that we will discuss here in a second. So there are several approaches to metagenomic and viral sequencing, targeted, amplicon, and whole DNA and RNA. And these types of data produce very different types of results that we'll talk about in a second. So targeted amplicon sequencing, you can see that they're here together, is really amplifying signal or information that is contained in specific regions. Those regions could be designed for a particular type of a pathogen, or they could be specific to a gene that can help us understand taxonomic relationship. When we talk about whole DNA and RNA sequencing, this is a completely different approach that produces different types of data that allow us to try to reassemble what is in the data, either by mapping on a particular set of references and looking at abundances, or trying to understand what novel information might be there that we are not capturing from a reference database, but still could be assembled based on an assembly of longer and longer pieces of genetic information because of the way that we observe that those pieces connected together in a number of different reads. So we'll talk about that in just a second. But these types of methods are used for different types of purposes. For example, you can see here an application of genomic sequencing to track viral outbreaks. For example, if we already have identified what is this outbreak caused by, what type of a pathogen, we can amplify specific viral sequences and then look for specific types of changes in the viral genome. Let's take one group of viruses, a family of viruses like coronaviruses, and we can start asking the questions, well, how is the data that we have in this type of sequencing different from a reference genome of that particular family or a member, a species of those viral families? Now, we can also look at all of the data that is available. So sequence all DNA or RNA, align the reads to a virus reference on multiple references, for example, a whole database of references. And we can now look at what's called metagenomic next generation sequencing, which allows us to study all of the genetic information in those in that sample. Here you can see some of the applications. So applications are detection of a specific microorganism, detection of specific mutations or changes in gene expression because of antibiotic resistance being developed in a group of viruses. Again, if you think of that community of viruses or bacteria, they are not a single identical member replicated many times. This is a community that has some diversity. Oftentimes, one of those members could become resistant and eventually replicate to cause resistance to be sufficient for the cells to not be able, or the body not to be able to deal with them. We can also look at changes in the microbiome, and those changes themselves could be a result of a condition or could drive the condition. They could increase risk of susceptibility. They could also change the way that our body metabolizes and deals with different types of challenges in the environment. And we can also study host response. So we can look at changes in transcriptomic or RNA sequencing to identify genes and pathways expressed as a result of infection and in the course of infection as well. Now, this type of data is produced by sequencing machines. These sequencing machines include short-read sequencers, and we will primarily in this program deal with short sequence data. So this includes short reads that are generated from the full genome or whatever sample we have. And we also have what's called long read sequencers. So these, for example, PacBio can be as long as tens of thousands of nucleotides. When we talk about short read sequencing, they could be as short as 30 nucleotides long. So very, very different types of data. And the challenges and the type of data that they produce are also very different. So let's talk about that a little bit. When we have data coming out of the sequencer, it comes in multiple types of formats, and we'll refer to these formats in all throughout the program. So 3 formats that you have to remember at this point. FASTQ, which is information about sequencing reads, and those sequencing reads will include the actual sequence, as you can see right here, the length, and also quality. So quality for each one of the characters, how confident are we in the quality of that particular nucleotide. FASTA files are files that contain references or full assemblies. So when we talk about a full genome, we're typically going to find that genome as a sequence in a FASTA file. And then also importantly for the references, we will have what's called the genome transfer format. So GTF file or GFF file. These files contain information about genomic elements. So where do they start, where do they end, and what do we know about them? So this is annotation for the FASTA file formats. What do they look like? Here's an example of a FASTA file. You can see that the FASTA file typically contains the name of whatever that file is about. So this is a Spodoptera frugiperida isolate, SF9, scaffold 12. So the scaffold is like a piece of the full genome, whole genome shotgun sequencing. Here you can see another example, rabies virus, the complete genome. If we look at the FASTA file from a human genome, we will see there that file divided into chromosomes, so divided up into pieces. Sometimes we can get a FASTA file with multiple genomic sequences, and they will have these kind of separators between the actual sequences. So this is a FASTQ file. The FASTQ file contains information about reads. So here we can see the read. Please tell me, what is the length of reads that you observe in this FASTQ file? Hint, this number is in the file that you see on the screen. So what is the number? Okay, excellent, right. So we're starting to learn how to read these files. You're exactly right. So here you can see the read itself, and here you see length 160. So just making sure that you guys are paying attention. So when the process of sequencing happens, we take genomic DNA. That DNA is broken up into short pieces, and that could be anywhere between 30 and 300 base pairs. This is an example for a specific type of sequencing. But here then we have library adapters that are placed in a flow cell. These flow cells, based on the machine, can be of very different sizes, and so they contain a lot of variation in the number of reads that we can sequence. And then we have this complex chemical process of PCR bridge amplification to produce enough material for the machine to be able to read it. And the way it reads the machine, reads the sequence, is by looking at image analysis. So we have different colors for different nucleotides. As they attach, we can observe them. And then we have these images, and that's where this quality score is coming from. The quality score is from the analysis of these images to give us an insight into how clear was that signal. So as a result, we have this long sequence. As the process of sequencing happens, we have these long sequences coming out, and the sequences are in short pieces. So when we get FASTQ reads or FASTQ files out of a sequencer like this, shotgun sequencing, one of the major challenges is to then annotate, to understand where did that particular read come from. If we have a long sequence, the sequence is already assembled. For example, we're looking at a particular pathogen and using Sanger sequencing, the challenge is then to align to other sequences and try to find variation. So here are the two main approaches to analysis for these types of data sets. One example for FASTQ files is analysis based on mapping, and FASTA files is analysis based on alignment, multiple sequence alignment in this example, to understand relationships. Here it's to detect variants. So two different approaches, and we'll talk about them in greater detail. Now, when we have this genetic sequence, what is the genetic or genomic sequence? What are the letters in there? The letters are called nucleotides, right? What are some of the letters that you know that are in nucleotide sequences? All right, excellent. A, T, C, and G. So we have 4 nucleotides. These 4 nucleotides form what's called amino acid sequences. So really, when we look at this kind of a sequence, we can convert. All right, look, Athanasios also knows the full name, so excellent. So we have here a lot of people that have this down. So you can see here that we have the nucleotide sequence, and then we have these reading frames. And there are several options for these reading frames. So we can get different types of amino acids based on the changes that we observe. So a typical process is to map these nucleotide sequences into codons. So who knows what are codons? I'm sure that Athanasios knows, but maybe other people know as well. Who knows what are codons? All right, so combination of 3 bases that code an amino acid. Babis, thank you, and Athanasios, yep. Okay, so these are triplets of nucleotides. Each combination represents an amino acid. So when we look at the changes in those nucleotides, we're expecting different types of changes in the amino acids. And so what you can see here is an example of an analysis where you have the nucleotide sequence mapped into codons. So you will have a 3-letter for each one of your letters here. So that means that these 3 make this one amino acid. And this codon mapping follows specific rules. So we know what the different types of amino acids are coded by, what types of codons. And there could be a lot of variation, right? So there could be a lot of different changes there. Now, importantly, when we think of functional implication of a nucleotide change, we are thinking about a nucleotide change that produces a change in the amino acid. And so there are multiple different things that we can expect from those changes, including a stop codon that will stop the translation of the sequence, and it will result in a shorter protein. And also it could be a change in the amino acid, and that change could be either in a specific given position or it could carry on, so we will have a deformed or a completely changed protein as a result. Okay, so let me pause here. I see some questions. Can you explain what is frame shift in reading translated amino acids? Well, a frame shift means that, as you can see right here, so this is the reading frame, and you can see that the result of different changes means that the sequence itself of amino acids that are being produced could be changed completely. So if you have a specific sequence and you change one letter, that letter could change the way that downstream reading happens. Or it could stop it completely. That's called the stop codon. Shift frame mutation means that it will change the subsequent reading of the nucleotide sequence, leading into different types of amino acids. So does that answer your question, Mona? All right, so now we can think of how can such a change happen. So one of the questions that we can ask is, if we align all of these sequences to each other and we observe some differences, for example, here you can see G and here you see T, if I observe such a change, or if I observe such a change in terms of the amino acid sequence, it means that the protein that is being produced from the sequence is different. This causes some potential change in phenotype. That phenotype could be defined in, we're talking about viruses, in its ability to replicate in different organs, in its ability to enter cells from different hosts, in its ability to be highly transmissible, so transmit more effectively between different individuals in a population. So it's a change in phenotype. And so oftentimes the question that we can ask when we do this multiple sequence alignment is, what is the relationship between the observed species and their ancestors? And the way that we can visualize the relationship is called a phylogenetic tree. So we can look at the evolution, the process of change over time, evolution, and we can ask the question of how are they interrelated? And that phylogenetic relationship really means that we assume that they have a common ancestor, and that ancestor then divided up into multiple branches and clades, eventually resulting in the species that we observe. So we can reconstruct that phylogenetic tree by using multiple sequence alignment. What type of information can that give us? Here you can see an analysis of multiple samples of Ebola viruses. So we have a number of sequences from Ebola viruses from Gabon, Liberia, Great Britain, and we can ask the question, how do they relate to each other? So if that relationship is closer, so for example, these are closer to each other than these, and what is their relationship in time? So looking at the x-axis, we can consider evolutionary time. The process of getting such information from sequencing data is alignment, conversion to amino acids, and then what's called a phylogenetic tree reconstruction using multiple sequence alignment based on multiple types of models. And we'll talk about that in greater detail. Sometimes we're not interested in the relationship itself, but we are interested in a particular change, a polymorphism in the case of population, or a variant in the case of a specific genome, and that mutation or a change can characterize a particular group of samples or an individual sample. Now, importantly, this type of an analysis is based on alignment of reads to the reference genome. So we can have, this is an example of a single sample. The single sample is aligned to the reference genome. And if it is aligned to the reference genome, we can very confidently identify some changes because they are present in multiple reads. If they are present in all of the reads, we will have high confidence. For example, here, you can see a change in this position. We have 1, 2, 3, 4, 5 changes. We don't have any other data that will indicate otherwise. well, I wouldn't say that this is a new virus. This is some kind of variant that emerges because most of the information is the same, right? So if we have a lot of similarity, that means that it is the same virus. It's just developing new adaptations. If we see a lot of these, and I'll give you several examples here in just a second, we can say that maybe it's a new virus completely. So we'll talk about that in just a second. Okay, yes, so important terminology here, haplotypes. When we say a new virus, we need to define if we're looking at different levels of taxonomy, species, family, phylum, etc. So we can talk about that as well. We'll talk about all of these concepts. But right now, what I wanted to mention is just where the data is coming from and what type of information are we getting out of this data. So in this particular example, we are seeing a change, a substitution, based on alignment to a reference. And what I wanted to point your attention to is that some of these variants have more confidence than others. For example, in this case, we have two sources of data, one read and another read. They do not match to each other, and they do not match to the reference genome. If they don't match, that means that we have low confidence. It could be a result of a technical error. So most of the time, this type of change is not going to be reported. This type of change will be reported as a variant. And then sometimes we also can have multiple regions, so multiple letters inserted or deleted. If we have such indication, these are called indels. So different types of analyses. One of the primary ways to characterize such data is what's called variant calling. And when we say variant calling, it's to distinguish between variants like this and all the other types of variants that we have low quality of information about. And we can think about how to compare samples or groups of samples to understand whether such a variant is present in all of the samples from a population or just a few. So a lot of questions that could be asked here. Let's take a look at a specific example. So here you can see a collection of reads. So each one of these lines is a read mapped onto a reference genome. Okay, so what is this reference genome? This reference genome is called beta coronavirus, Wuhan Institute of Virology 04, 2019-2. Okay, so this is the code name. This is where it came from. It's a beta coronavirus sequenced in Wuhan. You have all of these reads, and these reads are aligned to some reference. Now, that does not mean that these reads came specifically from this virus. You just take a sample and you're saying, okay, let me align it to this specific type of a reference. You will also have a consensus. A consensus talks about the majority of the nucleotides from the aligned reads, and they could be very different from the reference that we are using for alignment. We're just using this alignment to kind of give it a framework to follow. As a result, we can get multiple types of genomes, assembled genomes. And these assembled genomes could have specific variations that help us characterize those haplotypes or completely new viruses, right? So in the case of this coronavirus, we can talk about these are samples from bats, these are samples from humans, these are samples from China, these are samples from USA. And this is how they are related to each other, by these specific mutations, by these insertions and deletions, or by these sections that are completely different, right? So then we can start asking different questions about how they are related to each other and what could that mean functionally. So to kind of give you an insight into all of this practically, we have this multi-omics platform that we will use in this program. So in the program, we will not only speak about these examples, we'll actually do these examples together. So when we talk about the specific samples or the specific sequences and how they relate together, we will do some analysis. So before I get to your questions here in just a couple of minutes, let me give you an overview of the types of bioinformatic analysis pipelines. So here what you can see is an example of a pipeline. How these analysis pipelines could be done on the T-BioInfo platform to identify phylogenetic tree, reconstruct the phylogenetic tree, to measure gene expression variation between groups of samples, to align genomes to a reference to call variants, structural alignment, which will give us an insight into what does this alignment mean, and variant call, so detect variants that are specific to a group of samples that are different between each other. So in this program, we will talk about, in different sessions, about sequence variation, evolutionary relationships, functional inference from such difference, and the response from the host. And that gives us a good overview of host-pathogen interaction, but we will go into other examples as well. So let's talk about how we can do alignment, multiple sequence alignment. So let's go to the platform. Please follow with me to server.t-bio.info. And I hope that this will also give you some answers to some of these questions. Okay, so this is the link. Please let me know once you are here. Okay, for those of you that do not have an account yet, you can easily click on this access button and request a demo account. For those of you that do have access, you should have received an email this morning or last week if you register, depending on when you registered, to get access to this platform. We will use this platform extensively throughout this course, and we will talk about how you can analyze also data that you find on your own to process and to analyze this data. All right, if you arrived here and you are logged in, please put in 1 in the chat box so we know how many of you are already here. I'll just give it a couple more minutes so that all of you are here. All right, excellent. Thank you very much. So again, what we will do is alignment of sequences, just to understand what will that look like. So here you can see phylogenetic tree. And we will also use this section, and we'll do this here today just so that I can talk about some of these other ways to study data from here. So please go with me under virus quasi-species, even though the same methods are available in phylogenetic tree. We'll look at virus quasi-species. And we'll go to this first demo, virus multiple sequence alignment and phylogenetic analysis. So here you can see a combination of multiple types of use cases for different types of data. For example, you can start here and analyze data using multiple sequence alignment. So multiple sequence alignment is done on FASTA files. You have to have assembled sequences. You already know what the full sequence is. You want to align them to each other. So multiple sequence alignment essentially aligns the different sequences that you have. And here you can see some examples of different types of beta coronaviruses aligned to the Wuhan reference sequence. And then convert them to amino acids that have different physicochemical properties that we will talk about. You can also analyze how the different types of codons, you can see that each change could be a result of changes in different positions in the codon. So we can look at the different preferences over which positions we are incorporating. And then we want to reconstruct a phylogenetic tree. So we'll look at phylogenetic trees coming from multiple methods. These multiple methods will produce small variations or significant variations in how the relationship is organized and how the time, the evolutionary time, is reconstructed. So we can click on the tree space to study the variation between different results. And then let's click on end. And you will see here download pipeline output files. Okay, so how many of you were able to create this pipeline? if you were able to create the pipeline, please put number 2 in the chat box. All right, thank you, Ruthie. Anita? All right, so for Amy, again, I'm showing here on the screen, you go from start, multiple sequence alignment AA, select codon position MSA, and then all phylogeny methods, tree space, and end. All right, so you can review the video afterwards and try to replicate these steps. The important part of this is that you should get some of these demo example results. So here you can see multiple PDFs. These PDFs are going to be of the phylogenetic tree. And this phylogenetic tree is produced by 4 different methods. 4 different models, or 3 models for beast and 1 model for ape. You can also see a final kind of tree, and you can also see the actual output table, which aligns all of the data, but does not tell us how it's related, just an alignment. Importantly, we can visually interrogate such data. So this specific output right here, you can open it in Excel, but it's a fairly large data file, and we'll talk about how to deal with these data files. But I want to briefly visually illustrate what this data contains in terms of information. So please go with me to this link. This is the direct output from what we just saw. It's a sequence navigator that we developed to visualize such data, and will give us a sense for how these different types of sequences are related to each other. So the data set I'm going to be using is this one, comparison of the birth-death coronavirus. So you don't even have to follow along necessarily right now, just as long as you know that that output file could be visualized in such a way. What we see here is how thousands of nucleotides, so this whole reference sequence is about 30,000 nucleotides long for the beta coronavirus, or for the SARS coronavirus, about 30,000 nucleotides altogether. When we align the reference, when we align all of these samples to the reference, all of these are FASTA files to the reference, we can observe how many positions are changing from the reference in a bin. So here we can change between bin sizes, for example, 250 or 500 nucleotides. And we can start seeing that some of these are significantly different from the reference. So the more red they are, the less gray they are, the more different they are from the reference. And we can identify whole sections on the genome that are significantly different in a given protein producing gene. So here you can see a gene that is not protein coding, and here's a gene, the blue one, that is protein coding. This gene happens to be the surface glycoprotein, or the spike protein. The spike protein, and we'll talk a lot more about the spike protein, is responsible for cell binding to the cell receptors and entry of the virus into the host. And we can see that there's a lot of variation in this gene between the different groups of samples. Now, these groups of samples are organized by the type of coronavirus that these samples represent: beta coronavirus, COVID-19, SARS, and MERS, so obviously SARS-CoV-2. And we can see that according to what you see in this visualization, which group is the most similar to the novel coronavirus that causes COVID-19. So which other group, MERS, SARS, or beta coronavirus, is the closest to this virus that causes COVID-19? Okay, so Abir is saying MERS. Any other guesses? So again, the question is, which group of samples is the closest to this group of samples right here for COVID-19 based on sequence similarity? Okay, so Amy is saying SARS, Anita is saying SARS, Athanasios is saying beta coronavirus, Upasana is saying SARS. Okay, so again, gray means it's identical. So you can see this is 0 changes from the reference genome. So here we see all of these are gray. Probably leads us to think that the reference that we're using here is a beta coronavirus or the SARS-CoV-2 genome. Now we see here, this group, SARS, has the least differences, right? These are 0 differences, so whole sections are actually identical to here. And some of these contain a smaller number, so smaller in the tens. If we go over to the MERS, this has hundreds of changes. This has hundreds of changes. So it's obvious if you look at this image that SARS is the closest to COVID-19. That's why SARS-CoV-2 is closely related to SARS-CoV-1, which is the SARS section right here. We can visualize the way that they are interrelated by building a phylogenetic tree. So this phylogenetic tree, you can see it's annotated: beta coronaviruses, MERS, COVID-19, and SARS. And you can see that this clade, right, so this and this, represents a common branch from the main phylogenetic tree for COVID-19 and SARS. So SARS-1 and SARS-2 are the closest to each other based on a comparison between these different types of viruses. Does anyone have any questions about this? All right, if you have any questions, please post your questions in the chat box and we'll address them in just a second. The second type of analysis that we'll briefly describe is variant calling. Different types of data, remember that this is FASTQ files, not FASTA files, right? And here the objective is to identify specific positions that are very different from the reference genome. And these could be a result of selective pressure, variants and mutations that are associated with a phenotype. So let's take a look at an example. Here we have the Ebola virus. So we can ask 2 different questions from this data. One question is, let's visualize this data, okay? So if we visualize the data, we are expecting to see something that looks like this. And here we will find specific mutations, specific positions in multiple samples that all agree that there's a change from the reference genome. So somebody previously had a question about reference, or sorry, coverage. Right here in the gray, you can see the coverage. Right here on the bottom, you can see the actual reads. So if we just visualize our data, we can look at the reads, but we can also ask, well, altogether, how many reads are aligned to each position, which gives us a lot of different confidence measures for how we can be confident in a particular change. So if we do variant calling, we can identify specific proteins that have very defined mutations in groups of samples. These groups of mutations can help us characterize changes in the protein structure and function. And if we find those changes, they could be critical for design and development of vaccines, because we know that vaccines are only as good as the type of a virus structure that they represent. So that's an important thing to remember. And we'll talk about that in a lot more detail in the future. Now, the question here, doesn't matter which tree building method is used here. Obviously, that does matter. Let's take a look at what these trees actually tell us. So here's an example with Ebola virus again, and let's go to a website called Nextstrain. I'm sure that most of you have probably already heard about it. So Nextstrain is a place where you can look at different types of relationships based on data and some information about what this data represents in terms of where it was collected, etc. So you can see that there's a repository here on SARS-CoV-2, other types of viruses tuberculosis, Ebola vaccine, Ebola viruses, etc. So here you can see a number of different resources, and we can go to this link for Ebola virus. Okay, and this is a community project from a variety of groups that have collected samples from different locations in the 2013-2016 West African Ebola epidemic. So this tree that you see represents the diversification of the viral genomes as the virus spread between these different locations. Okay, so if you just play this animation, it shows you how samples from different locations and the associated number of deaths or infection from these different types of in different locations has caused a change in the viral sequence. Now, if you scroll down here, okay, let's do reset. You can see in which proteins on the reference genome did these mutations accumulate. So let me ask you a question. Out of all the proteins that you see on this reference genome, which protein do you think has the highest diversity measurement? This is the y-axis, the diversity measurement. So which protein has a position that is the highest in terms of its diversity? Okay, so Mohammed is saying SGP. So this is SGP right here. Anyone else? All right, so some of you think SGP, some of you think L. I think L is the correct, because you can see here, if you hover over, it says entropy 0.357. Okay, here you see entropy 0.285. So 0.357 is more than 0.285. So the correct answer is 319, and here's 385, so 357, right? So this is the highest one. The L protein, yep, it is difficult to see, but this just gives you some insight into how important it is to select the correct data and the correct analysis for what you are trying to do. So again, if you play this and you pause it, this changes, right? So where you sampled and how much data you have, this is this window. Within this window, your results could be completely different. So some of you mentioned SGP, nobody really mentioned NP, but you can see that in this case, this position is the highest in VP35. And so at this point in time, what was known is that the difference between these groups was best characterized by mutations in this position in this protein. So you might have thought, okay, well, this protein is the most important. As you continue playing this and you pause in a different moment in time, that information will change. So here, NP is the highest, right? So really the data is only is very critical for the analysis and for interpretation that we will do. So this is just an example, but I think also it's important to remember the significance of the data, the type of data, and the type of analysis. So we'll talk a lot more about the Ebola virus, these particular mutations that we are observing, the types of analyses that we can do with what kind of data, and even discover haplotypes, and not just the specific mutations, but actual haplotypes that are different. And we'll talk about the significance of this type of viral diversity studies that could be an impact on the phenotype, on how fast the virus spreads, how severe is the disease that it causes, and what are the results from such studies that have an impact on antiviral drug design and vaccine design. Any questions? Okay, so we're already running out of time. So let me briefly introduce you to two other mentors for this program, Dr. Harpreet Kaur and Dr. Mohit Mazumdar, will help you get the most out of this program. So we will have a number of practical exercises running these types of analyses, analyzing the data in R, looking at the relationships between different types of samples, and we will look at how you can develop a project of your own. So we will see how these examples that we present in the course of this program are really designed to introduce you to data repositories and analysis methods for you to develop your own independent methods and thinking about bioinformatics analysis. So let me pass it on to Dr. Harpreet Kaur. She will just briefly introduce her background.
Thank you, Ilya. So, hello everyone. My name is Harpreet Kaur and I have a PhD in bioinformatics. My research is on omics biomarker discovery for diagnosis and prognosis of liver cancer. In my current role, I am OmicsLogic bioinformatics trainer with Pine Biotech. I'm looking forward to being your mentor during this bioinformatics for infectious disease program and help all of you to learn about data analysis, machine learning, and visualization in the application to large data sets used in the biomedical research, especially in infectious disease. I will be happy to assist all of the participants in designing an analysis of the research projects or any kind of analysis. So now I would like to pass this session back to Ilya. So over to you, Ilya.
Okay, thank you, Harpreet. So again, we will have a number of technical sessions where we will run analysis together, look at the results. We will also introduce some of the examples in R, and Harpreet will be guiding those sessions and answering your technical questions. So let me now pass it on to Dr. Mohit Mazumdar, who will talk a little bit about project mentorship. Mohit?
Thank you, Ilya. And hello, everyone. My name is Mohit Mazumdar, and I'm one of the global representatives of Pine Biotech. In my current role, I'm working with scientists and researchers and students to assist with their omics big data research and collaborating with universities to provide support for bioinformatics training and education. Bioinformatics for an infectious disease is a special program. During this last summer, we had this unique opportunity to work with experts in data science, COVID-19 research, Ebola, malaria, tuberculosis, and many life-threatening diseases, and where we were trying to find answers to critical questions. So during the last program, we worked with our participants, and throughout the program, because of that resulted in more than 50 independent bioinformatics research proposals and projects. So the projects proposals were then peer reviewed by experts, and later the video presentation was recorded by our participants on topics such as different versions of human reference genome in RNA-seq analysis, GR37 and GR38, and their influence on in vitro studies of COVID-19 drug test on human organoids. Then another project was about genetic predisposition to acquire a polybasic cleavage site for highly pathogenic H1N1 hematoglutinin virus. So there were several research proposals that we worked through during our last program. So with this introduction, I would like to pass it on to Ms. Beepsha to take you further. Thank you.
Thank you so much, Dr. Mohit, for that overview, and to Ilya as well. So before I start giving you all an overview of the different resources, I wanted to talk to you all about what OmicsLogic programs are. So the goal of our OmicsLogic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. In our OmicsLogic programs, we will combine online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective. As you can see here, so we will have sessions like we did today. Participants will have access to courses, as well as we will be able to track all kinds of participant progress throughout this program. And I will shortly demonstrate this for everyone. Together with our collaborators, we have been building a growing online community of bioinformaticians in training, ranging from high school students to faculty and researchers in top universities around the world. Our team offers big data analysis tools like the server T-BioInfo platform and the SeqNav that we showed today. In programs designed around various topics, we discuss bioinformatics approaches for processing, analysis, integration, and interpretation of multi-omics data from oncology, neuroscience, agrobiology, and infectious diseases. I encourage you all to sign up for free on one of our portals and start learning or teaching bioinformatics. It is one of the best ways we can explore the world around us at the molecular level with zero equipment or lab costs. So now let us look at some of the resources that we will get as part of this program. I'm sure most of the participants already have received an email from my end regarding the resources, and also they are familiar with it. So this is what we called our bioinformatics organization page for all of our programs. So for the Bioinformatics for Infectious Disease program, this is your organization page. And here we will have the list of all the events mentioned. So today we are on 9th November and we have covered the topic of next generation sequencing, viral genomes in host transcriptome, and we have answered questions. We still have a lot of the questions in chat, which we will be answering in a few more minutes. And the next session would be on multiple sequence alignment and phylogeny. If you have not yet signed up, you can still sign up for this program. And you see here in this page, we will be able to see the progress and the activity of all the members who are joining the program. Now, before I move on, I want to show you what a profile looks like. So Anita, I'm borrowing your profile for an example because Anita here has a completed profile. So if you are a participant or whether you're joining us for the first time, I would request you to complete your profile. So what does a complete profile looks like? It looks like this, where you have a profile image and you have your full name mentioned. So when you sign up, probably this would just be your username. So you can change it via your profile and using the edit button that you will get after you click on profile. And you can also add your social media links. So the profile is helpful because as you saw that, we collaborate with our program participants on different projects, and then these projects are also sent for peer review. So this way, members in our community will be able to learn more from you. You would be able to collaborate with each other. And even if you're not joining this program, you can still have the opportunity to go out and explore and talk and network with people in the community. So that is the reason we would want you to have a complete profile. And also having a complete name will also be marked on your course certificates, which you get after completing the courses that are there on the platform. And now moving on, from the organization page, we will also be having access to the forums. So as you see here is a topic posted on the forum called session recordings. So this session will again be posted there. So we will have various topics on different ideas where participants can communicate with us even beyond these live sessions. And then you have access to asynchronous courses. So some of you may already have taken the Introduction to Bioinformatics course. And if you have not yet taken this course, please go ahead and take the first basic course, which is free for everyone, as is Introduction to Genomics. So you will be able to find them as well under here, Introduction to Bioinformatics. So all participants will have access to these different courses as well as projects. So you see here is one project on Ebola. And as Ilya mentioned, we will be discussing these in depth in the upcoming sessions. And for all participants, we will be able to see everybody's progress right here on the tab. So today, as everyone is joining, we do not have a lot of data from everyone because we are just starting the session today. But as we move forward, we will be seeing that there will be several different graphs of these colors where we will be able to track your progress across all of our platforms. The pink is suggestive of the OmicsLogic platform. The server platform is denoted by the gray, and yellow is for the courses and activities that you do right here. And now moving on, the other things that the participants will have access to is the server T-BioInfo platform. Now I know that a lot of people have requested the demo access to the platform. So the demo access to the server T-BioInfo platform will only give you access to demo pipelines. You won't be able to upload your data or do any of the hands-on analysis, but all participants should have access, educational access to the platform. And this platform is also available for research access. And if you need more information on that, please email us at marketing@pine.bio. I'm just putting my email down in the chat again. And Harshita, I'll just get back to your question in one more minute. The other platform, which Ilya also mentioned a while ago, which was practicing in R and Python. So we have courses on the OmicsLogic platform where participants will be able to utilize this platform for a browser-based experience. I'm sorry about this. Browser-based experience of utilizing R and Python for training and learning from these data sets that we discussed today, and also from other data sets, and also the SeqNav, which we had seen today. So these are some of the resources that are there as part of the Bioinformatics for Infectious Disease program. And we'll of course talk about the other resources, like we mentioned, some of the public domain repositories, which we will be mentioning throughout this program, and utilize those data sets for our own projects and see how we can learn about viral diseases. And to answer Harshita's question, the training certificate. Okay, so Harshita, for the training certificate, we have a parameter which we call getting 10,000 points. So you do not necessarily have to complete all of the courses listed there. We hope you do. And once you have over 10,000 points, I'll show Anita's. So Anita has been in a few programs and she has over 24,000 points. So she has received certificates of excellence in the past. So if you have over 10,000 points, you receive a certificate of excellence. If you have anything below 10 points, we give out a certificate of completion. We hope that you go through the different resources that are available and the different courses which we make completely available to you for the program. So I have a question from Mona right here. The question is how to maintain our membership so we will be able to use these programs beyond the 2nd of December. To answer this question very quickly, there are several membership options that you can take. And I think that once we go to the end of the program, we can send you the different links that we have to the membership options, so that you can keep on your access to the different resources for the program, or the resources to your access to the server platform. So each time you do complete a course, Harshita, so the question is how much points do each course carry? So the points that each course carry is 500 points. So once you complete a course, you get 500 points. And getting to 10,000 is not at all difficult, because you will not only be doing courses, you'll be running pipelines that gives you 50 points again, each of the pipelines that you run, as well as the OmicsLogic platform will also carry points. So it should not be very difficult. The only thing that you would have to do is keep on maintaining this and doing the courses at your own free time. You don't have to do it along with us, but you can do it at your own free time during this 1 month program and utilize these different resources. Now, I would want to pass this on to Ilya. Ilya, back to you.
Thank you very much, Bipsa. So as you saw, we presented here what we can learn about viral and bacterial pathogens by analyzing their genomes. We talked a little bit about next generation sequencing, which is key to this kind of ability to analyze and ask questions of the data. And we talked a little bit about some additional resources that if you see gaps in your knowledge, for example, if you are not familiar with some of the terminology that we use today, you don't understand the reads, you don't understand coverage, you don't understand how this data is prepared, we ask you to use some of the synchronous resources to catch up and be ready for this program. Some of the data sets that we will analyze are fairly big. So you will see data sets that are gigabytes in size and hundreds of megabytes in size. To analyze and process all of this data on your own computer can be prohibitive just because of the size of memory and CPU, and your pipelines will just take forever. So to make this program go faster, we've prepared this online resource called the T-BioInfo platform. We'll also introduce you very quickly to advanced methods of analysis. You don't have to wait and build the pipeline. The pipelines are already pre-built. The results are already informative as you will get them. And then finally, this community that we have allows you to interact with mentors, peers, work together on projects, ask questions, make sure that you start getting into the more interesting and advanced questions much faster. So what we'll do in the next several sessions is go over sequence analysis, alignment, phylogenetic trees, analysis of genomic and transcriptomic data, and you'll start seeing how some of these publications that talk about viral phylogenetic relationships, resistance to antibiotic drugs, and also relationship with the immune response and disease progression could be answered using data sets that are already available on NCBI and other publicly available resources. For those of you that have already joined the program, I have an assignment. So for those of you that are just joining and you're still not familiar with many of the terms that we're using, we ask you to complete Introduction to Bioinformatics. Actually, this is also for those that are joining for the first time and you just want to try one of these courses. This is a course that is free. You can complete it in probably under 1 hour. And what it has is an overview of different types of important terms and concepts and how bioinformatics is used across a wide range of applications. Importantly, it also has a survey that helps us learn about you and propose some meaningful coursework or programs that are upcoming. So really a way to personally address some of the interests that you might have. This type of material has been reviewed by faculty, undergraduate, and graduate students, and all of them agree that this information, even though it might seem a little bit introductory, is useful even if you are a professor or a researcher, and especially if you're a student. So it's an introductory course, but can provide an interesting level of detail. We also ask you, if you're already done with Introduction to Bioinformatics, to complete a course on metagenomics. This will give you some insights into what types of data could be extracted from sequencing data, We have a course called Introduction to Metagenomics, which again is free, or Metagenomics 1 if you're a part of the program. And for a project, we ask you to look at Spotoptera frugiperta, which is a project that allows you to analyze sequencing data and detect a novel pathogen to characterize it and understand why it might be important for biologics production pipeline for biotechnology companies. So again, your assignment is to look at Spotoptera frugiperta, complete Introduction to Bioinformatics, Introduction to Metagenomics, or Metagenomics 1. And also we have a blog post that talks about virology resources for those of you that are interested in virology and how to analyze virology data. Please take a look at this blog post that will allow you to explore some of the databases and some of the tools that are available in these different repositories. So this is the link to here. Okay, let me answer some of the questions that I've seen here. Okay, so the question, could you reference T-BioInfo platform is used in publications? Yes, we have a whole link to a list of publications for different algorithms. Many of the algorithms used on the platform that are public domain have been used extensively on a variety of different projects. So we have a list of those publications. We'll be happy to send you a detailed view of what it does. So please email us marketing@PineBio, and we'll get back to you with any additional questions. I wanted to summarize by sharing a profile of someone who is successfully completed the project and some of the feedback that we got from that person, because I think many of you might be wondering, what is the difference between this program and some of the other online programs that you might get in data science and bioinformatics, maybe in molecular biology or virology? So I think that what we've seen is that many of the activities combined together really develop in an experience that is not similar to many of these programs. We don't take just general concepts and go through the methodology. We guide you on how to ask questions about data and answer them using analytical tools. The objective of the program, and for those of you that really want to succeed in the program, is really to develop an independent project. So there are several reasons why you might choose to be a participant in the program and benefit from all of these resources together. The analytical platform, the coding platform, the educational asynchronous courses, and the interactive sessions that we will have. And so Beepsa just posted a link to this blog. You can go through the blog and just kind of see some of the reasons why the people that really benefit from the program have joined and how it's different from some of the other online resources. Also, whether you're a part of this program or not a part of this program, we welcome you to join our groups on LinkedIn and Facebook. We post there regularly resources, free data sets, example projects, as well as opportunities for career development on these groups. So if you are a budding bioinformatician or you're an expert in the field, we want to be connected in the long term. So please join our groups on social media. That will help us stay in touch. And I do want to mention that this program is going to continue for the next several sessions. We will have a special recording of this session made available, and we'll be happy to share it with you if you've already registered with an email. And I'm happy to answer any final questions before we finish this session. Okay, well, if there's no questions right now, I was excited to get started with this program and hope to see all of you join us again next time. Thank you for registering, and we are looking forward to working with you on developing your own independent bioinformatics projects in the next few weeks. We'll have this recording shared with you by email. Anyone that wanted the materials, you can review that in your own time. And if you have any additional questions, please feel free to reach out to us, Again, marketing@pine.bio. Thank you again, and we will see you again next time.